Okay, I think we should go ahead and start. Thank you, everybody, for being here in the room or online. It is my great pleasure to introduce to you today Dr. Darren Browning. So Darren has been presenting in this forum for a while now, so many of you know him very well. So Darren has earned his bachelor degree and master and PhD in cell biology from the University of Toronto in Canada before moving south, I think, running away from the cold to California to the Strip Research Institute to do his postdoc. After that, he came here as assistant professor in the Medical College of Georgia. And he is now a full professor in the Department of Biochemistry in Augusta University. Darren has really a keen interest in education and teaching. He has been awarded Distinguished Teacher Award by the graduate school, and he was the director of the Biochemistry and Cancer Biology graduate program for many years, and he also ran a very nice program, the Augusta University 3 Minutes Thesis Program. This is a pretty good thing, actually. It's an excellent idea. Darren has published many papers, more than 60 papers in high-impact journals. He has been funded by the NIH, and he is currently funded also by some small business technology transfer grants to start his own company that he actually gets also some funds from the Georgia Research Alliances to do that. So he will tell us a little bit today about the drug development that he is doing in his lab and hopefully give us some insights about how to start a company. Thank you, Dan.
Thank you, Ahmed, for the introduction. I don't know that I'm going to tell you about business, but we can discuss later maybe whether it's a worthwhile endeavor or not. So anyway, thank you all for being here as well. It's always both a privilege and a pleasure to share some of the things that's going on in my laboratory with my colleagues in the Cancer Center. What Ahmed didn't tell you is that the title or the topic that I'm going to discuss today actually came about because of a conversation that we had in the parking deck a few weeks ago when we're talking about our efforts and aspirations for drugs or small molecule drug development. And I know that there's several other laboratories in the Cancer Center that are also looking into this. So I thought this was a great forum to share my experience so far in drug development, and hopefully some of you that might have some more experience than me can give me some feedback. So the first part of the talk, I'm going to be discussing the rationale for why I would be interested in developing gut-targeted phosphodiesterase inhibitors in the first place. And then the bulk of it, the latter half, is going to be on some of the progress we've made in actually coming to our current stage and the future aspirations that will move through to drug candidates that we're actively excited about right now. All right, so I need to make or AU requires me to disclose that I am co-founder and CEO of a startup company in the incubator space over in the CA building. The company's Target Biotechnologies, and we're interested in developing gut-targeted therapeutics, but this is not a CME requirement for disclosure because we're not selling drugs, we're not making money, there's no financial conflict here. So why would I be interested in developing gut-targeted cyclic GMP phosphodiesterase inhibitors? And so these phosphodiesterases cleave cyclic GMP. So if you inhibit them with a small molecule, then the cyclic GMP level increases. And so there's a lot of information, both in the clinical realm and in the preclinical realm, to suggest that if you increase cyclic GMP, then it can be used to treat or with cancer prevent a variety of important diseases. And And so for those of you that have been around for a while, mostly at the RIP seminars, I talk about some of the details of my colorectal cancer prevention work with phosphodiesterase inhibitors. And everybody in the room is certainly aware of the importance of colorectal cancer being, the top 8% of all cancer diagnoses and deaths internationally are colorectal cancer. And so our work has shown that phosphodiesterase inhibitors can reduce the risk of colon cancer development. Also, inflammatory bowel disease. Now, this is people that have this, then it's not a lethal disease, but they are at higher risk for developing colorectal cancer. And, at least for moderate to severe forms of colitis, they really have a miserable life. So there's a lot of morbidity associated with IBD. Increasing cyclic GMP, there's evidence from my laboratory and other laboratories preclinically that it can be therapeutic. Chronic constipation. Currently, there are drugs that elevate cyclic GMP that are prescribed to treat chronic constipation. And this one, again, is not a lethal condition, but it's so prevalent. It's estimated that up to 20% of the Western population are affected by this. And when we get to the elderly population, it's up to 70%. And these ones, again, they have significant effects on quality of life. And so lastly, increasing cyclic GMP can also affect the epithelial barrier in the intestine. It's not currently something that's used in the clinic, but there's a lot of interest in understanding this because it's associated with a variety of diseases. But even more relevant to our own campus, where inflammaging is one of the foci that we're investing in. Leaky gut allows bacterial contents to enter the or enter into the tissues, and this can lead to inflammation. And so it's thought that a leaky barrier that arises in the elderly can drive inflammaging and a lot of the associated diseases such as Alzheimer's and Parkinson's and whatnot. And so increasing cyclic GMP can also affect, or it can increase or augment the epithelial barrier. So before going forward, I just wanted to refresh everybody's memory on the intestinal structure and function. So most of the digestion and absorption of nutrients that we eat ends up in the small intestine. So what's passed to the large intestine or the colon is largely deplete of nutrients. And so it's passed there. And then there's two functions. This is full of a robust microbiome that can ferment some of the complex carbohydrates and fiber that are small intestine is incapable of digesting, and this provides additional nutrition, but also a lot of the digestive juices that go into the digestion process are reabsorbed in the colon. So these are two major functions, and this generates, of course, fecal material. So if we take a closer look, just go into the colon a little bit, we have this outer serosa connective tissue. Immediately underlying that are circular and longitudinal muscle layers that are used to propel the contents through the lumen. And then just beneath that a little bit further is the submucosa. And this is highly innervated. There's a submucosal plexus as well as a myenteric plexus. There's more neurons in the gut than there is in the brain. So this is called so-called gut brain. Also, this is where the vasculature is. If we move in a little bit further into the mucosa, this is there's a lot of cells, including immune cells in the lamina propria, but this is really largely this epithelial. And so the epithelium forms a very important barrier between the luminal contents in the bacteria and these underlying tissues. And so the surface you can see is also has this protective mucus barrier, and that's secreted by these goblet cells. So these are differentiated cells that emerge from the crypts when the differentiated cells are at the surface to perform their function for a few days, and then they undergo spontaneous apoptosis. And so they're replaced by cells coming out of the crypt, which is necessary because without replacing these apoptotic cells, then there'll be a barrier breach. Bacterial contents will come in and incite inflammation. And so down at the base of the crypt are stem cells that give rise to progenitor cells that then proliferate. And as they move up out of this proliferative stem cell niche, which is provided by these myofibroblasts that secrete specific factors. As they emerge from this, then they differentiate in this transit amplifying zone. And then, as I said, it takes a few days for them to get to the surface. They last for a few days. So the whole gut typically is recycled, the surface anyway, in 5 to 7 days. So where does cyclic GMP come in? If we look at the colonocytes, the epithelial cells, whether in the base of the crypt or on the surface, they all have this the cyclic GMP function. They express this receptor guanyl cyclase known as GCC, and this is stimulated by endogenous secretagogues. These are peptides, guanylin and uroguanylin. When they bind to it, it activates a cytosolic guanyl cyclase activity that generates cyclic GMP from GTP. And the cyclic GMP has long been known to activate type 2 cyclic GMP dependent protein kinase, or PKG2, and this leads to activation of CFTR, which is a chloride channel, which allows a salt secretion and then fluid matches. So this causes secretion. And there's actually 2 companies that have taken advantage of this secretion function of cyclic GMP. These are both peptide mimetics of the endogenous hormones, and then you give these to patients, and the secretion has been effective at theoretically flushing the system to alleviate some forms of chronic constipation. Also, as a side, and interestingly, it can dampen gut pain as well in IBS patients by an unknown mechanism. So my laboratory, as well as largely 2 other laboratories, the main one being Scott Waldman and Thomas Jefferson, mostly using genetic models, knockout mice, we've shown that cyclic GMP, in addition to the secretion effect, also can affect homeostasis. So this is this crypt luminal proliferation differentiation. And so activation or increasing cyclic GMP is thought to by, in a PKG-dependent fashion, suppress proliferation at the base of the crypt, as well as suppress apoptosis at the top of the crypt. And so, again, this was all in knockout mice. Maybe 10 years ago, my laboratory was interested in saying, well, if we take wild-type mice, can we increase cyclic GMP and actually suppress proliferation and apoptosis? And anyway, at the time, this was pre-FDA approval of these drugs, so it was difficult to come by. So my laboratory kind of looked elsewhere, and this is what got us into the phosphodiesterase inhibitors. Since Viagra and tadalafil, these have been prescribed for many years to treat erectile dysfunction. They're easily obtainable, and we found indeed that if we put these in a mouse, then it can increase cyclic GMP, mostly by blocking the degradation pathway for cyclic GMP. So effectively, these PDE5 inhibitors amplify the endogenous cyclic GMP machinery. And so just to show you what some of this data looks like, if we measure proliferation using Ki67, you can see there's a lot of proliferation at the base of the crypt. And if we increase cyclic GMP either by treating the mouse orally with sildenafil or with linaclotide, which is one of these synthetic GCC agonists, we get a suppression of proliferation at the base of the crypt. Also, if we look at the apoptosis and the luminal epithelium, this is actually understated here because when they undergo apoptosis, they're rapidly shed into the lumen. But so you can see also we get a dramatic inhibition of the number of apoptotic cells together. We also noticed increase in differentiation, particularly of goblet cells. It's quite obvious here. But we're thinking that mostly this is the result of this change in homeostasis, because if we're suppressing proliferation at the base of the crypt and also slowing down apoptosis at the top, and you can envision that of cells moving more slowly up through the crypt, so they're differentiating a little bit, within the crypt itself. Yes.
What's the treatment schema of these mice?
It's kind of like as soon as you give the drugs, we can put sildenafil in the drinking water and they drink it. So it's sort of almost linear by the days, and then when it gets 3 to 5 days, which is a gut turnover, it has a maximal effect, and it's stable until you remove the drug, and then it phosphodiesterase 5 inhibitor. This is Viagra, the main ingredients, and this is linaclotide, which is the agonist. So two completely different mechanisms of increasing cyclic GMP, which really adds credibility to the notion that this effect is due to increased cyclic GMP.
And were there any side effects in these mice?
No. And, people take these drugs, or at least tadalafil for benign prostate hyperplasia. They take it daily for years. And there are some side effects in people, which I'm going to allude to a little bit later. But in the mice, no changes in body. They're happy. In fact, anecdotally, they just seem to be just happier in general and more active. That's another story. So anyway, based upon on this finding, this sort of fundamental biological role for cyclic GMP, we hypothesized that if we're suppressing the number of proliferating cells at any given time, then we might be able to reduce the risk of colorectal cancer or prevent it. And the reason for that is because each one of these brown proliferating nuclei, they're subject to replication errors, and also to genotoxic stress, which is really a major influence in the colon, because we're concentrating some of these, the xenobiotics that we eat along with our food. Yep.
Another naive question. Mm-hmm. I don't know this, but I always thought that it also depended on what you're eating on the turnover of the drug.
This is something that, in my view, is really understudied. And so, in terms of chemo prevention agents for colorectal cancer, nothing. I mean, aspirin has been the billions of dollars have been spent on aspirin, and it's not really something that is going to be,
What I was alluding to was like in these mouse models, the mice are on a very regulated diet of what they eat. Do you think this should be repeated in mice with a high-fat diet and stuff like that? Because that's what the human situation is.
I think that's a great idea, but if you'll see towards it in the next few slides, it's ready for a clinical trial. So we're done with the mice, actually. At least that's what NIH tells me. They're not interested in more mechanistic studies. These aren't new drugs. These have been in the clinic forever. So, it's a matter of seeing if it works. But if I can continue, you'll see where we've gotten with that, actually. So, this was maybe 5 years ago, something like that. We used two mouse models for colorectal cancer. Both of them, it made a splash in the cancer prevention world, because we showed that physiological doses of sildenafil, and this one also linaclotide, were able to suppress polyp formation by 50% in both of these models. So again, I think that the notion that we were using a physiological dose, a very small dose of the drug, like half of what you would give an adult male for erectile dysfunction, and it was effective in these animals. And so this would, again, it made a bit of a splash, so that based on that, two independent groups, one in Sweden looked at the hospital discharge register, huge database of patients, and also somebody here, actually just across the river, Scott Sutton, he looked at the VA database. And they basically asked those that were taking, PD5 inhibitors, are they more or less subject to colorectal cancer? And they found, in support of my preclinical data, that up to 50% reduction in colon cancer incidence, suggesting that it really could be useful for prevention in people as well. So the next logical step is to take tadalafil, which has a good PK, and test this in the clinic prospectively to see. And so several people in the cancer center, including Dr. Cortez, some gastroenterologists, who worked for a good year trying to establish a protocol to submit to IRB, and it's kind of waiting on the gastroenterologist because it's going to be their patients. But it is sort of in progress. So as a PhD scientist, I don't see patients. I'm more interested in the mechanism. And so we use intestinal organoids to study, since we have this turnover, proliferation, and then differentiation, then apoptosis, There's no cell lines that can recapitulate that. But in the last decade or so, stem cells can be isolated from the epithelium, from the crypts, and we can culture them under, conditions that mimic the stem cell niche, and they form these what they call pseudoguts, where the crypt's at the base, and they have this common lumen. And we have numerous patient specimens, thanks to Ronnie Bolag and his biorepository people, and some really generous pathologists that helped to get some of these tissues. We biobanked numerous patients at different regions of the gut, small intestine, cecum, colon, different regions. So these are extremely useful models because we can maintain stem cell conditions and study stem cell and progenitor and proliferation. We can also change the conditions to differentiate them. We can study the differentiated, which is very important in some of the stuff we're doing right now when we're looking at barrier function. Or we can actually look over time, we can actually look at that transition, which would be the transit amplifying zone of the real colon. So very, very useful system. And just to let you know, the research in progress, the model that we're chasing right now is shown here, where we've shown using the organoids that there's a massive increase in ERK activity that occurs when a stem cell gives rise to a progenitor cell. Progenitor cell takes off proliferating, and that's largely due to this increase in ERK. And increasing cyclic GMP in a PKG2 dependent fashion is able to block this. And so based on other literature, there are some targets that we need to look at. I'm trying to get this project funded. So to turn the page, I also showed you some evidence that increasing cyclic GMP is capable of reducing apoptosis. So this is the spontaneous apoptosis that happens to the differentiated cells at the luminal surface. We investigate this a little bit further. So if we give the mice this drug called dextran sulfate sodium, it's toxic to the luminal epithelium. And so we put it in the drinking water. And this is what it looks like stained for tunnel after about 3 to 4 days. Massive apoptosis. Now, if we give these animals a few days before and then during the DSS treatment, we can really block that apoptosis. So it really is not just changing the homeostasis, it really is protecting the robustness of these cells to resist apoptosis. And so if you can imagine if you're breaking down the epithelial barrier like this, over time you're going to get a massive influx of contents into the underlying tissues, and you get granulocyte influx, and you get a massive inflammation. And this is shown here as the disease activity index, which is a combination of bleeding and diarrhea and body weight, body mass changes. And so we have to take it out after 5 days or it'll be lethal. But in that case, you see a massive increase in disease. But those taking vardenafil, we cut the disease index in half. So this is some data to suggest that it may be therapeutic for IBD, inflammatory bowel diseases. Interestingly, though, if you take these wild-type animals, over the next few weeks, they've actually come down again. After a month, they're completely normal, back to growing body mass, the increase in body mass, they're all happy again. But if you look at the gut, there's these long-term damage to the gut, so that one of the things that happens is it retains a leakiness. And so if we can measure this by gavaging a FTSE-labeled dextran, so this complex polysaccharide, which can't get through a normal healthy epithelium, but if it's damaged, it can leak through and get into the vasculature, and we can detect it in the plasma. And so that's what we're measuring here, FTSE dextran coming into the plasma after oral gavage. And these mice that have recovered from this inflammatory process have a leaky gut. Single dose of sildenafil can seal this up so that it's no longer leaky. And we don't know the mechanism. Is something that I'm interested in looking at now. But also, if we just look at the fecal water mass, so diarrhea effectively, we're also fixing that with a single dose of sildenafil. So it's a really remarkable effect on the epithelial barrier function. So again, as a PhD scientist, the project, the research in progress, or the model that I'm trying to look at is shown here, where we can increase cyclic GMP with these P5 inhibitors, and we believe that PKG2 can activate FOXO3A, which we've shown previously is uniquely expressed on the luminal epithelium. We've shown that it can upregulate antioxidant genes and protect against redox stress. Other people have shown that it can affect cytokines. So this is the model that we have, and we're testing now. Yeah.
Your data with the IBD is very, very exciting. Do you have to give the drug before the initiation of the IBD, or?
Well, that's the thing. So, I was just talking to Ahmed about, getting this funded by NIDDK, and it can be a challenge because oftentimes immunologists are on these panels and they're not so much interested in the epithelium as they're interested in the immunological result of that. But it's a good question. We haven't looked at that. There's several much better models so this is more an acute thing. Patients don't come in and then, I'm going to have IBD tomorrow. They come in with a chronic, and so a T cell driven mostly. So that needs to be done. That's an important experiment to do that we didn't do. Yeah.
Have you looked at if the treatment is affecting your gut biome? Is it affecting secretion?
I would put money on yes, for sure it is, because you just have to eat an orange instead of a steak and your biome changes. You can go for a walk versus sitting and your biome changes. So this dramatic effect on the barrier, because barrier function is known to, still microbiome is very exciting. It's been very exciting forever. So we've cataloged everything and all we know is important and it changes. The microbiome affects the barrier and the barrier affects the microbiome and the immune system's in there, but it's really complicated and not. So the fact that we're affecting the barrier, there's no question. But have I looked at it? No, because what do you do with the data, right? All right. So turn the page again. I know this is some research to suggest that these drugs are useful for treating constipation. So as I told you, GCC analogs or agonists are currently prescribed for this. But PD5 inhibitors are not, and so this is a bunch of experiments to do with undergraduate students. They come into the lab off the street with no experience and no knowledge whatsoever. So you get them in there, looking at constipation in mice, and very simple, cheap experiment. We gavage the mice with a charcoal meal, and then we just wait for the black fecal pellet to come out, and that's how long it takes. It's called transit to go right through the animal, and so in a young animal, like 6 to 8 weeks, it's like, starts around 2.5 hours and is over by about 4 hours, as shown here. And we can look at this and we treat these animals with sildenafil or linaclotide linzess, and we see no effect at all on transit. However, if we subject them to disease models, opioid-induced constipation, we give them loperamide. This is Imodium, the active ingredient Imodium. We increase the transit time, and we give either an acute dose of sildenafil or chronically over 5 days, and we can reduce the transit. This is a remarkable thing as well. This is clearly a neuromuscular effect of cyclic GMP that we're interested in pursuing. Also, a post-inflammatory, post-infectious IBS model. They also have transit issues, and we can similarly reduce or somewhat normalize transit. So while opioid-induced constipation is clinically important, post-infection IBS is not as important. But as I mentioned earlier, up to 70% of the elderly population suffers from constipation, and there's a lot of reasons for that. But probably a good 30% of them are due to chronic transit or issues. And so we're interested in looking at whether sildenafil and related PD5 inhibitors are able to treat constipation in elderly, in this case, mice. And so we have what's not considered elderly mice, but I showed you the 8-week-old mice. There's no effect on transit of these drugs, but if we actually just look at the baseline transit in older mice, so 7 months, 8 months, something like this, then they have actually a longer transit time. So they're effectively constipated. And this is already well established in rodents in the literature. As they age, much like humans, they become the slow colonic transit is an issue. Of course, I can't. So we treated these with sildenafil, and we're able to actually normalize it, just like in the other disease models. This is very exciting data. Anyway, so we have one program now that's we're trying to get funded to look at this in elderly mice. So we have actually now we just ordered is $400 for one mouse. that's 20 months old. So we have 10 mice in there. It's $4,000 for a couple of cages of mice. And so we're going to look at barrier and transit in these mice. So again, our model that we're chasing now is not intracellular, not PKG dependent. It's that we believe that the cyclic GMP increases, it's exported extracellular into the lamina propria, where it can interact, it can affect neuronal firing. Let's put it that way. This is not my idea. It was published 2013, so many years ago, by Stu Briley's lab in Australia, where he demonstrated that extracellular cyclic GMP can somehow impact afferent nerve firing. And so using our human organoids, we did show that actually this is cyclic GMP treated with linaclotide, sildenafil, or the combination. This is intracellular and this is extracellular. So there's a robust export of cyclic GMP into the medium. So this is a promising avenue that we're looking at. All right, so that's the end of the why I'm trying to make, phosphodiesterase that are gut targeted. Hopefully I've shown you enough evidence to say that, well, yeah, there's some therapeutic value for treating constipation, colon cancer prevention being the big one, since there's nothing currently available for this. And then, of course, leaky gut and this inflammaging is an important potential as well. So the low-lying fruit is, well, why don't we just use linaclotide? This is the GCC agonist. This was a screenshot of their webpage. So it's currently prescribed for IBS-C, but you look down on the bottom here, and it has some problems in that it's contraindicated in children because it will kill the pediatric mice or juvenile mice. They end up getting dehydrated, and this is separate. I can discuss that later if interested. But even in the patients, the ones that work, a lot of them are non-compliant because they have, bloating, and the most common reason for non-compliance is that they get chronic diarrhea. So for a cancer prevention modality, this is just not going to really work in the clinic. At least that was my thoughts based on the side effects. But recently, in September, Scott Waldman's lab, he presented some clinical data to demonstrate that in humans, if you give these things orally, they just don't get to the colon. So there's no bioactivity in the colon. So again, for colon-based diseases such as cancer, these aren't going to be useful. So he actually called me out and said, yeah, this is the way to go. So we might not be the only ones looking at colon cancer prevention with PD5 inhibitors in the clinic. All right, so why don't we use then current, available PD5 inhibitors? And so these things are designed, like most orally delivered small molecule drugs, to be rapidly taken up in the small intestine predominantly, and they go through first pass the hepatic portal system to the liver before going out into the circulation to get to their target organs. And for the PD5 inhibitors, it's likely for erectile dysfunction and benign prostate hyperplasia or BPH. But then they're systemic, so they go to all these other organs as well, and that leads to side effects just based on the efficacy of the drug. But there's also a lot of drug interactions shown here, nitrates being the most important one. So people taking organic nitrates such as nitroglycerin for coronary artery disease cannot take PD5 inhibitors because it'll result in a synergistic hypotension. And so this is actually lethal. So big warnings that you can't take these. So this systemic delivery is a problem with respect to drug interactions in the least. So again, it may not be perfect for at least for colon cancer prevention, where they will be taking it daily. So since our target tissue is actually the inner lining, the colon epithelium, it just seems ludicrous to design a systemic drug that goes in systemically. So we decided to try to make a drug that didn't go systemic, that was targeted to the gut epithelium. And so how do we do that? If you look at some of the most common PD5 inhibitors on the market today, they are designed, like most small molecules, to be lipophilic. You don't have to be an organic chemist to look at these things. So yeah, they're hydrophobic, and they're designed specifically that way so they can cross through plasma membranes to get into the vasculature. okay, is necessary to reach the target tissues. And so our approach to generate gut-targeted PDE5 inhibitors was to take advantage of the prototypic PDE5 inhibitor sildenafil. Why? Because there's so much literature and in the chemistry literature to let us know that we can replace this methyl group on the piperazine ring, which is hydrophobic. And the idea is that we can put a more polar group on there to give it a harder time getting across the plasma membrane. So maybe it can still get into the epithelial cell, but it won't go further. So that was our idea. And thanks to a collaborator and actually co-founder of Target Biotechnologies is Irina Lebedzieva. She used to be faculty in the chemistry department. She's since left Augusta University. Very talented synthetic organic chemist. She made more than a dozen compounds. It's much easier for the chemist to make a compound than it is for us to test it. So that, I had to develop in my laboratory, sort of a pipeline that would allow us to eliminate certain ones that aren't going to move further. And just in a nutshell, the way we did that was just to make sure that some of the analogs that we made still had activity for PDE5. We're also interested in looking at to make sure they're specific to PDE5. So we looked at some other ones. We looked at their ability to cross the membrane and increase cyclic GMP. Yep.
Now, when I sit in, I don't do drug screening.
Mm-hmm.
But when I sit in study section, I hear people talk about the reproducibility of the assay. What did you have to do to get the assay, activity assay?
This one here. If you even just Google PDE assays, there's a dozen of them that come out. I didn't develop that one, but I tested a few, and one of them works pretty well. It's based on fluorescence polarization to be able to detect the cleaved cyclic GMP into GMP. So it works. So that was one. This here is basically 2 years in the making, this whole scheme. And out of the 12, we have 2 now that we're working because they fell off of the pipeline at some point. So, again, making sure that they can affect the drug target. We wanted to make sure that, they were designed to be less permeable in the membrane. So we looked at that in vitro. And then this is really not pharmacology, belongs in the PD slot. But before moving forward, wanted to make sure that if we give it to an animal orally, that it was still able to increase cyclic GMP in that luminal epithelium. Those that pass that, then it gets a little bit more expensive, which is why we did it in this order, is that we look at the ability of an oral dose to get into the bloodstream. And we lost a couple of compounds because of that. They did show up. And then, lastly is, do they have any function in the mice? So measuring proliferation, barrier function, transit, and colon cancer. And we're currently actually working on whether our lead drug candidates that have passed all of this are able to affect colon cancer. Another thing that we're into now is toxicology, and there's the toxicology where NIH is not so willing to fund an academic grants when you're just looking at toxicology, because that really goes into the realm of drug development to move it into the clinic. Yep.
So are these existing PDE5 inhibitors that you're working and modifying them?
Yes, we use sildenafil as the parent structure, and we modified just that one group.
But you're adding something else.
Yes, I'm going to show you right now, actually, if you want to have a look. So this is the overall scheme to put the data I'm about to show you into context.
And do you compare it to sildenafil?
Yep, you'll see the data. And so,
with questions that are set.
No, this is, again, I'm new to the drug development field, and so this has not been a straight line. This has been moving back and forth and trying to figure out what works. And I welcome all and any feedback. So this is the sildenafil is a parent compound. We know this is the business end that fits in the active site. This is just some hydrophobic. So we changed this one to give you an example of some of the many compounds that we've looked at. Malonyl-sildenafil, this is like my baby mostly, is a carboxylic acid group derived from malonic acid on there. So this big negative charge should definitely impede its ability to get across the plasma membrane. Veronal-sildenafil, it's polar. It's not obviously, it's not charged in that way, but there's a really interesting compound I'll talk about as our second lead. Amino-sildenafil, there's a positive charge on that one at physiological pH. So the first thing, and I'm showing on the right here, is the activity of these things in terms of their ability to inhibit PDE5 activity in vitro. And so sildenafil is shown here in the circles. Milano sildenafil has almost identical IC50. Boronal sildenafil is a little bit less potent. Putting that amino group on there really made it less potent, but it's still a candidate because it's sub-micromolar efficacy, so it's still decent. But definitely modifying this with a positive charge did affect the IC50. So really, based on this data, Milano sildenafil was the one that we moved further with along the pipeline. Yep.
With these drugs out there, most of these companies have already made all these a lot of modifications. I'm sure that they're running into some patent issues and all that.
Well, we already have a patent pending, and so I'm excited to see what the reviewer says. But we have this pipeline developed now, so actually, that's got some value. We also, in these dozen compounds, we know what's going to work now. There's a fine line between limiting its ability to get across the membrane and then not getting into the cell at all, versus getting in enough, but it doesn't go further. So we know now, we have a lot of information that I'm not going to show you about what it takes to get through. So that has some value. If the reviewer comes back, the patent reviewer comes back and said, we need, there's too much prior art or what have you, we can make another one. at this stage, with again, Irina is a talented medicinal chemist. So anyway, so there's a whole family, many families actually, of phosphodiesterases. I showed you data for PDE5, but there's related PDE6, also cyclic GMP specific. PDE9 also is an important one because that is also in the intestinal epithelium. Different enzyme kinetics. There's a bunch of them that are cyclic AMP specific and also ones that are dual specific. So we looked at several of these. This is the Milano sildenafil data. PDE5, it's the same as sildenafil. PDE6, this is actually sildenafil. So actually by putting that carboxylic acid on there, it's actually increasing its efficacy, its ability to inhibit PDE6. So this is maybe a concern, but PDE6 is only expressed in the retina. And so patients that are taking erectile dysfunction drugs at the higher dose range, meaning 100 mg per kg or more, or like 100 milligram pill, or more, they experience visual things. And this is because of this cross inhibition of PDE6. However, for our drugs, they hopefully don't go systemic. So in order to get into the circulation, they need to cross a bunch of membranes. To get into the retina, that has to cross more. So I'm not really concerned. I'm actually excited because it expresses, it actually has submicromolar or micromolar activity against PDE9. So the inhibition of PDE9 and 5 may have a more robust effect. Really didn't have any significant activity on the dual specificity ones that can cleave also cyclic AMP. So the next part of the pipeline is, okay, we put this thing in, it still has pharmacology. Can it cross the plasma membranes of cells? So we developed, and this is another thing that adds some value, is so we developed this cell line that actually expresses all of these things, the GCC and the PD5, PKG. And so we use this as a model. We give these cells just a little bit of linaclotide, so it generates some cyclic GMP, but it's kept to almost nothing or undetectable because of PDE5. And so if we put a PDE5 inhibitor on these cells, if it can cross the membrane and get into the cells, we can measure the cyclic GMP. We can also measure the activity of PKG downstream because it can phosphorylate a substrate that we can see on a Western blot. And so if we just look then at sildenafil, and this is sildenafil, I'm showing the increase in cyclic GMP at different doses, and then boronal sildenafil. So these are very similar, but the two that we put, this is Milano, the negative charge, and then we put the positive charge. And so they're shifted by almost 100-fold. So definitely they can still get into the cells, increase cyclic GMP, but it takes a lot more. So it's consistent with the actual, our design of the drugs. And so this change in efficacy of the drug to get into the cells is reflected by phosphorylation of VASP as well, in that it takes, about 10 to the 4 to get what, 10 to the 2 can do of sildenafil. So it's compatible with what we see here. So these assays for membrane permeability are done in a Petri dish, a tissue culture dish, just naked cells, but that's very different from what happens in vivo that has this big mucus layer and bacteria. So we asked, can we actually get this drug into and to increase cyclic GMP in the actual epithelial, luminal epithelial cells. And so we gave these mice a single bolus dose, and then several hours later, we actually harvested the epithelium and looked at cyclic GMP content. And as you can see here, we actually looked at the ileum, the terminal part of the small bowel, as well as the proximal and distal parts of the colon. And across the board, we were able to get significant increases in cyclic GMP, indicating that this drug has no problems getting across the mucus into the epithelial cells. So, of course, the big question is this drug now able to get into the bloodstream, or is this stop in the epithelium? And so for this, we needed to get a mass spec fingerprint, and thanks to Wembo Z in the proteomics mass spec core, he's been extremely helpful in helping us define these things so that we can measure the levels of these compounds in the plasma. And so this is the data shown here. This is a dose curve where we would gavage either sildenafil or melanosildenafil, and we've taken plasma. And we took plasma 1 hour later because this is where sildenafil is known to peak. And you can see that the melanosildenafil is just undetectable in the plasma on this scale anyway, where sildenafil increases even to the micromolar level. So this was exciting for us because we've shown that we can increase cyclic GMP in the epithelium with these, but it's not getting into the plasma. But at 1 hour, if there's some kind of resistance to getting into cross membranes, it's possible that there may be a delay in the plasma entry of the melanosildenafil. So we did a time course, and we got the same has been published umpteen million times for sildenafil, but we really don't see significant amounts of melanosildenafil, a second or delayed peak of melanosildenafil, suggesting that we actually have a gut-targeted non-systemic PD5 inhibitor. Interestingly, if we actually collected the fecal pellets from these animals and extracted also the compounds from them, and you can see dose-dependent increase in the amount of these drugs showing up in, this is a 4 to 6 hour period of fecal pellet collection, and a dose-dependent, increase in the melanosildenafil is showing up in the feces, which makes sense if it's not going systemic, where sildenafil, it just doesn't show up at all. Not that we expected this sildenafil to, because it's heavily modified. And then over time, this has really excited us because in these young mice, usually the black pellet, the transit time is 4 hours, but even 8 hours, and that's as far as we assayed, it's still being shed in the feces, which means it's in the colon for an awful long time. So this was not part of my pipeline, but before moving into some of the efficacy in terms of affecting colon cancer or transit or whatnot, we wanted to make sure that there's not some kind of bioactive metabolite being metabolized in that intestinal epithelium, leaking into the circulation that might be giving us, results that are systemic. And so I hooked up with this one. How do you measure blood pressure of a mice, right? You put a cuff on it. Actually, people do that. They put it on a tail. But Mike Brands is in physiology is an expert in this. And so he has people that can, surgically implant a probe into one of the carotids and then have like this remote transmitter he puts under the skin. And so the mouse is sitting there, you turn on the transmitter with a magnet and the thing is running around and you get real-time telemetry of heart rate, blood pressure, and movement. So we did this and we treated the animals with an organic nitrate. This is isosauroid mononitrate, which is a clinically prescribed drug. And so what do we got here? So this one here is just the nitrate by itself. If we put in the nitrate with sildenafil, you see this massive drop in blood pressure. But if we put it together with melanosildenafil, we don't see this drop in blood pressure, which makes us a little bit more comfortable with the notion that there's no bioactive metabolites entering the bloodstream. And then this, this lack of effect is shown quantitatively here. So we give this animal to mice, gain in the drinking water. We measure it for over a week, and there's no change in body weight. And we just do a quick histochemistry with an H&E. We don't see any major damage, no edema, leukocyte infiltration. So we're confident that the drug isn't really hurting the mice. We didn't see any other problems with it. So we gave it to them for a week. And we use two different methods to look at proliferation in the colon. The first one here is more objective, just flow cytometry of the whole colon using Ki-67. And again, melanosildenafil is able to, actually more effective than sildenafil at suppressing proliferation. If we just want to count PCNA positive nuclei, again, sildenafil and melanosildenafil are both able to suppress proliferation. Yep.
So how does measuring the blood pressure correlate?
The organic nitrates that are used to treat coronary artery disease, they're vasodilatory. They one of the best known functions of cyclic GMP is smooth muscle relaxation. And so to open up coronary arteries that are occluded in heart patients, they give them nitroglycerin, which releases nitric oxide, and it activates cyclic GMP production by activating a soluble form of guanyl cyclase. Does that make sense? Well, the thing is, if you have nitric oxide release, it's causing activation of a soluble guanyl cyclase. And if you're also inhibiting the degradation of soluble of cyclic GMP, now it's synergistic. So you're going to get a massive systemic vasodilation and a precipitous drop in blood pressure. Does that make sense?
I'm just thinking like there could be other metabolites that don't affect.
Yeah, it's true. I mean, that's something that we're looking at now. But if it has a bioactive PD5 inhibitory activity, we would detect it, and we didn't. So it's a good question. All right, so this one, the melanosildenafil, again, we're very excited about it. At this point, it is a drug candidate, and I'm funded to look at it and see whether it can prevent colorectal cancer now in a mouse model. I wanted to, in the last few minutes, talk about the boron-sildenafil. This is actually Irina Lebedeva's baby. For some reason, organic chemists love boron, boronic acid. And so, shown here, again, we did the in vitro assays against PDE5, slightly, but more or less same as sildenafil. There's no increased effect on PDE6, which is nice, but there's not really an effect on PDE9, and it doesn't affect, again, the dual specificity in phosphodiesterase. Interestingly, again, we put this more polar group on there, and actually it seems in the cell-based assay, it's more effective at getting in and inhibiting PD5 than sildenafil, the parent compound. We don't understand this. And so, and even the VASP data supports this. It's just as efficacious as sildenafil at getting across the membrane and having an effect. And so, one would ask, why are we interested in this in the first place? Is not clearly it's getting into the cells very easily. And the reason for that is because boronic acid is capable of making these reversible covalent bonds with these hydroxyls. So picture this as being carbohydrates. So boronic acid is capable of making transient covalent bonds with carbohydrates. And so if you look in the animal or in the tissues, this mucus layer is composed largely of proteoglycans, and the glycosaminoglycan parts are carbohydrates. So the idea is that maybe this boronic acid part of this molecule is able to interact and with the mucus layer, and even after the luminal contents are gone, that it might actually form have this reservoir of drug that can feed the luminal epithelium for a longer period of time. So that was the reason that we continued looking at it. I tested the drug, and again, gavaging, we get increased cyclic GMP and all the different parts of the colon. It's actually very an impressive higher effect in the ileum than melano or sildenafil. So we also got a signature for this one, and we looked at it in the plasma, and we got almost identical results as melanic acid, which is really intriguing. So sildenafil, again, this is an independent experiment. It goes up with doses. We did not detect, and you have to understand that this dose here, this is even with allometric scaling, without allometric scaling, this is like 100 times what you give a human. But with allometric scaling, this is, for mice, this is 12 times the human dose equivalent. This is a huge amount of drug, and it's still not really coming in. So it's an interesting compound. Again, we look for a delayed peak. It's very, very small amount, but we don't see any delayed entry into the plasma either. So we look at the feces, and again, the shapes of these curves is very similar to monosildenafil in that we're getting a dose-dependent increase in the feces, and also over time, it's being shed in the feces for a long time. The error bars are very large on here because, interestingly, in this experiment, there was a difference between male and female mice. So we're actually going back and have taken a second look and actually have the samples. We just have to give them to Wimbo.
But also the sildenafil being released into the plasma.
Yep.
Expect less difficult,
Well, in the feces, so again, in this case, so I should have had the line. So this is you don't see it ever go in there. And in fact, more than 90% of sildenafil enters the plasma within like 30 minutes to an hour. And it's first pass metabolism, it's mostly gone, it's turned into desmethyl-sildenafil, and then it's conjugate. I mean, I don't expect it to get into the feces anyway, but you need to control, so. Because otherwise I'm showing there's nothing, Does that answer your question? So the last piece of data that I'm going to show you today is we also did an experiment with, and this is just flow cytometry. Again, we give ronald sildenafil to the animals, and we didn't see any change in body mass or behavior. But when we look at proliferation in the colon mucosa, we see that it's pretty much the same effective as sildenafil, monosildenafil at suppressing proliferation. All right, so almost the end of my talk. And so where are we in terms of drug development? It's a long and expensive process. We've been playing around in here to develop compounds, but the pipeline has produced these two that I just showed you. We have a couple of others as well but we're moving these other two forward. We've been most of what I've shown you today is this pharmacological and efficacy and pharmacokinetics. We're now moving into toxicity studies, and then it's like, well, what should I do for toxicity studies? And so the NIH is happy to give you this large, a document that describes all of these expected toxicologies, but it's very generic. So what they would expect from our drug versus a cancer drug that's IV is a very different thing. So we're kind of feeling our way here. But to sort of take the minimal amount that could be summarized here, these so-called ADME studies, absorption, distribution, metabolism, excretion. Some of them I've done already. We looked at how much would it cost to actually outsource these experiments, and so I got a quote of about $350,000 just to do the basic stuff. And so that's one of the reasons that we have a company that's doing it for a fraction of this cost. And so based on all of these, we're focusing now on metabolism. So in response to one of the questions about other metabolites, also all of our work is done in mice. We need to go to another non-rodent vertebrate. And so the metabolism studies will identify that one. Also looking at transporters, if these drugs can inhibit some of the multi-drug or the drug efflux pumps, then they could create toxicities. And so these are at least the minimum low-lying fruit of toxicity. So we're doing these now. Just to show, we'll take our drug and then just look, this is not our data, this is just how we're going to do it. Put our drug in with actually microsomal fractions from we're going to use epithelial microsomal fractions from the gut epithelium of different species to try and find one that's similar to humans. We've got so many, plates full of barriers right now, Capo-2 barriers, which is what FDA requires to look at transport. And then once you've got transport, like apparent permeability, then you need to look at which transporters it might be inhibiting or being used. We're also using our organoids to put on these filters to do barrier studies, because with the Ronald drug that I showed you already, it seems to probably require this mucus barrier that's not associated with Capo-2, which is actually a cancer cell line. Interestingly enough, if you do a quick PubMed search, there's a paper published just 6 months ago or what have you that actually showed that organoids grown in 3D and then put into a 2D filter are actually better I mean, here they say there's the same, but they're actually better because they have a mucus than CACO2. Unfortunately, I don't think that the FDA is ready to take data from organoid filters yet, but hopefully this will grow because, you know, I think that CACO2 will end up just showing one of our compounds going straight through, whereas in vivo, it doesn't behave that way. So ultimately, the organoids, I think, will be better. All right, so that's where I'll finish up. A lot of the biological data, the rationale for developing the drugs was done by graduate students in my laboratory over the years. Most of the drug development data that I showed you has been done by me personally. I get my hands dirty. And also some talented undergraduate students. Last year I had 4, I got another 5 coming in this summer into the lab. They get involved in this and be co-authors on papers. Is really good. I have collaborators in our department for various things. I can't stress enough that, you know, this drug development work absolutely requires collaborations with medicinal chemists. And for anyone who's interested, Abed knows we're recruiting right now medicinal chemists in the chemistry departments, not in MCG. Wembo Z, of course, all the mass spec stuff he did, he saved me countless hundreds of thousands of dollars. Nevin is the one that did the blood pressure. Fuyong and Megan in cellulology, they're involved in aging. In a few months, I'll be doing another talk on all of our aging data. Ronnie, again, wouldn't have the organoids to look at the mechanisms without his assistance. I have 2 groups of clinical investigators, so Amel Sharma and Satish Rao. They're very excited about looking at PD5 inhibitors for constipation, also Parkinson's. So, you know, we're working towards an R1, hopefully in June, to look at that. And then Dr. Cortez and the gastroenterologist, Ken Vega Erickson, we've worked, again, we have an IRB protocol ready to submit. It's just a matter of the PIs of this to move forward with it. And then the funding. And so with that, I'll take any remaining questions. Thank you. Yes.
Yeah, just a quick comment from the immunology perspective. If you want your grant funded by NIDDK, use the Fiona Powery model, the RB high transfer into RAG. They will accept that. DSS does not have.
Yes and no. I've submitted that one. I got Kumar Ranakasamy, who's published, you know, even methods for that model, and still didn't go. You know, I may come and see you, and maybe you can help bolster it. I appreciate the feedback, though. Yeah.
Any more questions? Okay, great. Thank you, Darren. That was a great talk.
Yeah.